My name is Katherine Drotos. I'm a PhD candidate in the Department of Integrative Biology at the University of Guelph, and I'm excited to talk to you about some of the questions I've been looking at for my thesis. I would like to begin by acknowledging the land and the land protectors that make my work possible. I am fascinated by lichens and the ecosystems in which they thrive, and many of these ecosystems are threatened by human exploitation and encroachment. The resiliency of these lands and the lichens that grow there is a testament to how incredible they are, and to the people that protect them. I would also like to acknowledge that the land on which I work and live is the ancestral and treaty lands of the Attawandaron people, the Mississaugas of the Credit, and others, and I recognize that this is Dish with One Spoon territory. The Dish with One Spoon covenant describes the concept of sharing the land and its resources for the mutual benefit of all. We can think of this as a mutualistic symbiosis with the land, creating a balance that enables the whole system to survive and thrive through time. Symbiosis is typically defined as when multiple organisms engage in a long-term, stable, and intimate relationship. Within this, there are subcategories: mutualism, where both parties benefit; commensalism, where one party benefits and the other receives neither benefit nor harm; and parasitism, where one party benefits and the other is harmed. For the purpose of today's talk, let's focus on mutualism. This strategy allows for the exploitation of niches and often drives co-evolutionary relationships, where functions may be divided amongst the organisms in the relationship. For example, one organism may lose one function and upregulate another, while the partner organism does the opposite. This is often seen in symbioses with organisms from different kingdoms, where functional specializations may widely vary. Together, these organisms can perform functions and occupy habitats where perhaps neither would be as successful as an individual. A classic example of symbiosis is the lichen. Lichens can be loosely described as a symbiosis between fungi and algae, and are well studied at the ecological level, but recent research has suggested that there is much more to them than meets the eye. We now know that there are other organisms involved in the relationship, such as basidiomycete yeasts and bacteria, but their role is unclear. And even before we get to that, there are still so many unanswered questions about the lichen symbiosis, like how the partners share and divide functions at the cellular level, and why we can't reconstitute lichens in an artificial setting. One of the things that I find particularly interesting about lichens is that they are not a natural group, but are rather an indication of how adaptive fungi really are. Let me show you what I mean. This tree shows a simplified topology of Ascomycota, one of the two largest phyla of fungi. Each color represents a symbiotic state. Lichenized lineages are shown here in green. And so there are 5 lineages represented here in Ascomycota alone. Generalizing across all of these groups based on analysis of only a few, despite their shared morphology, could be problematic. It would be as if we generalized across all animals with wings, based only on a couple of birds and flies. Understanding the nature of this evolutionary pattern and why so many fungi become lichenized requires a wide-scale, comparative, and multifaceted approach. First, we need to know some of the basic fundamental characteristics about lichenizing fungi at the cellular and molecular levels. Things like how big their genomes are, how they're packaged, and how this differs between lichenized and non-lichenized fungi. Very little of this is known of lichens, and almost none of these parameters are studied across lineages. To inform the development of deeper hypotheses, we need this fundamental knowledge on their cellular and genomic biology. Essentially, we want to find out what makes a lichen a lichen. To answer some of these questions, I developed a methodology specific to lichens for measuring nuclear content. This was based on existing protocols, but required some modifications to work with lichen tissue. The main structure of a lichen is the fungal hyphae, which weave together to form mats in layers, and in bigger forms, this is called the lichen thallus. We found that the extracellular matrix and cell walls of the hyphae prevent the nuclear stain from reaching the nuclei, and the efficacy of the initial methods varied widely by species. We're not sure yet exactly what component is causing this, but this is definitely an avenue for further study. There are many parameters that went into developing this protocol that I won't go into here, but I'm happy to talk about it on Twitter or email if anyone is interested. The exciting thing about this method is it allows us to visualize many structures which are often difficult to see and measure in lichens, including nuclear content. So what is nuclear content? You might know it more readily as genome size, which is the total amount of DNA contained in one copy of the genome. It's also often referred to as C-value. Here, I'm using the term nuclear content because we aren't certain of the ploidy levels of these cells at this time. Genome size is a useful metric for looking at macroevolutionary trends across a group that shares functional traits. This is largely due to the strong correlation between genome size, and cell division rate. Simply put, as genome size increases, the size of the cell also increases, and so cell division rate decreases. This leads to cascading effects in higher traits, such as development and metabolic rate. Genome size measurement is also relatively cheap and fast compared to a lot of other genomic tools, so we can examine a wide breadth of taxa. So from what we know about genome size across eukaryotes, fungi tend to sit at the smaller end of the range. On this graph are 3 eukaryotic kingdoms, with fungi at the top in magenta, animals in the middle in blue, and plants at the bottom in green. Fungi do exhibit a bit of a spread, but it's largely clustered down at the lower end. Prior to this study, there were fewer than a dozen records for lichenizing fungi. So where do they sit on this axis? Down at the low end of fungi. They're pretty tiny. What I'm going to show you now is a snapshot of some of the data we've collected. Here we have nuclear content, or C-value, measured in picograms for 10 lichenized fungal species across 3 orders. It's worth noting that 1 picogram is roughly equivalent to 1,000 megabase pairs. All of the results here are quite small, very small genome sizes, but there are still some obvious differences. Both Lobaria species over here are slightly larger than the rest of the group, and the Phaltidra elizabethae, right here, is almost 4 times larger than the rest. Hold that thought, because we'll come back to it in a minute. Now we're looking at the diameter of the hyphae of these same 10 species. It's worth noting that there is little information in the literature about hyphal diameter across lichenized fungi. It seems to be only recorded for specific species during other types of study. All of these measurements here were taken from cells that were intact and viable prior to fixation to avoid differences caused by health of the cell. There's quite a bit of variation here within most species, and once again, Phaltidra elizabethae is quite a bit larger. We're not sure yet why there is so much variation within a species. It might be due to the age of the hyphae themselves, or where in the thallus they came from, though we tried to mitigate this by taking samples exclusively from the leading edge of a given lichen species. Hopefully, further study will lead to some answers here. Putting all this together in a phylogenetic context, we can start to parse out some patterns. On the left is a pared down phylogeny, or a topology, of the 10 lichen species. The Lobarias and the Phaltidra at the top are all members of the order Phaltidrales. Anaptychia crinalis, below that, is a member of the order Teloschistales, and the remainder are all in the Leccanorales, the fungal order with the most lichenizing fungi. There is some alignment of the trends of both nuclear content and hyphal diameter here, and we see a clear divergence between the Phaltidrales and the others. It will be interesting to see whether these trends hold as we measure these species across the various lineages, and what patterns emerge when they are compared against their non-lichenized sister taxa. Now for something completely different, a phenomenon we've noticed that doesn't appear to be noted anywhere in lichen biology. In almost every species we've looked at, there are distinct clusters of bacteria. When examined in detail, they appear to be held in place by a jelly or a membrane, as best shown in the bottom left photograph here. We don't know yet what these clusters are or what they're doing, only that they're consistent across most lichens. One hypothesis is that the lichen is somehow sequestering them, either for a beneficial purpose or for defense against them. Another is that this is just how bacteria behave when contained within the lichen thallus. Or it could be that the bacteria form up as a biofilm within the thallus to achieve their own purpose. This phenomenon will definitely require further study, and we are interested if anyone has ideas about this. In summary, through the design and optimization of this protocol, we now have a reliable method for visualizing and measuring a number of cellular and nuclear traits of lichenized fungi, and we can see that so far, their genomes appear to be quite small, even for fungi. We can also see that they have a somewhat variable hyphal diameter across species, and possibly within species, and this will lead to other questions and hypotheses about why that is, and why some species or groups may exhibit certain patterns. We've also found that many lichens contain bacterial clusters, and that remains an interesting mystery. Further work in this project and beyond will see us sampling as wide a taxonomic range as we can get, of both lichenized and non-lichenized fungi, to see what happens to the genomes of these lineages when they become lichenized. We'll be examining some of these traits in the context of lichen ecology, as this area is well studied and understood. For the methodology, we will be looking into further uses and adaptations to look at other structures that are typically hard to visualize and measure, and we will also be looking across our testing results to see if we can predict which species need further modifications of the protocol based on their position in the phylogenetic tree or another characteristic. With that, I would like to extend great thanks to a number of people. Dr. McMullen has been incredibly helpful with procuring samples and providing lichen ecological knowledge. Dr. Sbrabilla has been an invaluable fountain of lichen wisdom, and his lab is doing some incredible work with lichen metagenomics and study of the extracellular matrix. Huge thanks to Mackenzie Ruffo for help with data collection, and to my advisors and members of their labs for their feedback and support. Thanks also to folks who have shared data, funding agencies, and finally to you for listening. I'm happy to answer any questions or continue discussions via email or Twitter. Thank you.